Good evening to everyone. So we are back in the class, biology class. So I actually went through your papers, MCQ papers. What do you think? Marks are good? Marks are really bad. Bad in the sense that I like enough, but still you could have get more. I if I would have told about meiosis and mitosis, ne? Hari, come on, it's okay. But still, the highest mark is so far coming from Gaul. I don't know from this side who is the highest. I'll let you know when I go through the correct numbers. Hari. No one has passed 40 out of 50. Yeah. Okay. So unfortunately, that's very bad. Okay. So please improve yourselves. Go through the papers again and again once I gave it back to you. Make sure that you will be doing that, right? So practice is a must. Do more questions regarding meiosis, mitosis. Go through past paper questions and please go through that, okay? And find out more questions from school papers and all that also. And in like 1 month or like 1.5 months, I'll be giving you a past paper book also, first term papers. Once I'm done with certain content, I'll be giving a certain set of past papers together so that you can do it at home and we can discuss also some of the papers, not everything, right? Okay. And there's one important thing I wanted to tell you guys also, that is actually regarding the cases that we heard in recent days. You may have heard that one girl has jumped from Lotus Tower, and previously another one from Altias, and so many suicidal cases in young children like you guys, isn't it? So I actually wanted to say that so many things can happen, problems can happen. What we need to learn is how to face to them, isn't it? So you guys need to know how do you handle a situation. Sometimes like certain pressures are coming to us also. Now when you think at that moment, you will feel like I can't face to the society, I can't do anything, nobody would love me. Likewise we feel, but with time you may understand it is not a thing that to even care, right? Okay. So give time for things to sort, and also always reach to the like not to everyone actually. Sometimes the case may be worsen if you reach to some unnecessary people, but still you can trust your parents, you can trust your teachers or something, someone which is closer to you, or a trusted friend. Please talk, right? Talking will sort many problems. I'm telling you this one. I actually remembered you guys when I heard those stories. I feel very bad. If someone was there to listen to their stories, sometimes they may be still living, no? Isn't it? As biology students, you might know how it is hard to bring a life to earth, isn't it? So there are so many things other than loving and those affairs and all that. There are so many things to do, isn't it? So that you should understand you are a worth life. You can do a lot of things to the world rather than just having commit suicide, isn't it? Hari, okay. So we'll get back to the lesson, children. Are we having new one? Hi, Radha. Are you a new one? Okay. So, children, I have asked you to do some questions in the tute and come. No, shall we discuss the answers for that? Hari. So, the ones who don't have the tute, let me see if I can share the questions. Let me see. In the structured essay section only, I asked you to do the questions, right? Isn't it? Hi. 1 minute, we'll share the date with you. Tute in this no PDF is given, I'll share online. Others all have the tute now? Yes, guys? I'm going to the questions now. In this one, the first question is asking, write the photosynthesis in a simple equation, no, isn't it? Yeah, photosynthesis reaction in a simple equation. What are the ingredients of it? Carbon dioxide, 6 carbon dioxide gas along with 6H2O. On the arrow, what do we write? Sunlight, chlorophyll. Will give C6H12O6. What is this one? Glucose plus 6 oxygen. Harine? Right. So this is the equation that we are expecting you to write there. Right. Next question is asking what is photosynthesis. Let's write the given definition here. What is given there? It is a metabolic process into chemical energy. Hi, let me read the correct definition one more time. Others mark if it is correct, right? It is the metabolic process by which light energy is trapped and converted to chemical energy. Right? You can continue also if you want. You can add chemical energies stored in carbohydrates, fats, oils, and proteins. All right. Next question asking what type of metabolic reactions that photosynthesis belong to? Anabolic reactions. Right. Next asking what is the significance of photosynthesis? I'll be reading the answers. Mark if it is correct. All life on Earth depends on photosynthesis directly or indirectly. Did you write that? Now only writing the answers, no? Okay. Fulfill both carbon and energy requirements. Next one, provide oxygen for respiration of aerobic organisms. Next one, maintain oxygen carbon dioxide balance in the atmosphere. Next one, production of fossil fuel. Next one, maintenance of global temperature. By the way, if you are asked to do some homework, please do it and come, okay? You know, like at the beginning, there are so many enthusiastic people in the class, right? So at the end, you know, when it comes to results analysis, you may see that only few people will get A passes and only a few people will get B passes. Why is that? That enthusiasm is not maintained. That's the reason, okay? You have to work harder. All right. So next one, asking what are the types of photosynthetic pigments found in the higher plants? Chlorophylls and carotenoids. Next one, asking to define a photosystem. This is how you write this. Chlorophyll molecules, other organic molecules, and proteins are organized into complexes in the thylakoid membrane. Of chloroplast. These are called photosystems. Photosystem 1 and 2 definition is asking, you need to write this first part to define both of the photosystems, and additional thing is the photosystem 1 contains a special pair of chlorophyll A as the reaction center, which absorb which nanometer? 700 nanometer wavelength effectively. Arada. One is missing, that boy is missing. And next question, the same one, what you are changing here, phase 2 and 680 nanometer wavelength effectively. I'm not going to rewrite it, you can change it and write now. All others have done, no? Shyly. Very smart. Okay. Continue. Why? You forgot? Both of you. These questions are the ones which will make you understand the lesson, right? So we discussed the light dependent reaction of photosynthesis. These questions, when you are doing this, you will understand what happened there. Are you done? Shall I erase? Right and finish. Erasing. What is the cellular organelle which light reaction of photosynthesis takes place? Chloroplast. What specific location of the cell which light dependent reaction takes place? Thylakoid membrane of the chloroplast. Given below a diagram showing light dependent reaction of photosynthesis. Answer the following questions using the above diagram. What is shown by the Z-shaped pathway of light-dependent reaction? What is the answer, Putte? Linear electron flow. Next one. What is the major event which is happening in the photosystem 1 and photosystem 2 after absorption of light energy? Both. Common one is asking in the question. Excitation of electrons. That's the common thing, right? Number 3 is asking what replaces the electrons which went out from photosystem 2? P680. What replaces? High releases of electrons coming from high releases of water. Correct? Number 4. What replaces the electrons which went out from photosystem 1? Electrons coming from PS2. Isn't it? Do you remember the pathway? Should I revise it? No. Coming from photosystem 2, you have to mention. That is the specific answer here, right? You have to write specific answers here, right? That's why I'm writing also. Electrons coming from PS2. Right? Fifth question. There's a repetition. Yeah, there's a repetition. Cut that question. What are the end products of the light dependent reaction of photosynthesis? What are the end products with it? ATP, NADPH, oxygen. What are the molecules that store light energy as chemical energy in the light dependent reaction of photosynthesis? What's it? The numbering, there's a slight change. So this one should be fifth one, this one should be sixth one, right? Hi. So here, what are the molecules? ATP and NADPH. Okay, children? Next one. Which photosystem gave the energy for ATP? Which photosystem, children? Photosystem? Who says photosystem 2? Who says photosystem 1? If you can remember, I just drew the line, the circle from photosystem 1. The electrons are coming from photosystem 1, so it gives from photosystem 1. There's no like separate pathways at the same time. Simultaneously, both are happening even if we draw two diagrams, right? Understood? To make more ATP, some electrons coming to the primary electron acceptor of PS1 going rounds and rounds. Okay. Next one. What photosystem gave energy for NADPH? Again, PS1. Agree? Next one. What are the end products of splitting of water? Oxygen, H pluses plus electron. Next, last question is asking, what is meant by photophosphorylation? So how do we define? Synthesis of ATP using energy. Nah. Energy, photo means light, no? So using light energy production of ATP, we call it as photophosphorylation. If you can remember, this was there in a previous page where we skipped and come. Can you all come to that page quickly? Page number 50. Synthesis of ATP using solar energy in photosynthesis, we call it as photophosphorylation in the textbook, page number 50. Aridhu? What they say? Hmm. That is how it forms, no Pute? This is the correct definition, right? Okay. Found the definition, 50th page. Alright, before we go to 11th question, we didn't discuss the answer for the exam question. No, I told that there is an exam question came in the last recent most past paper. We didn't do that now. Alright, we'll write that question first in the writing book or in a paper. You may attach it to the book if you want. Because this answer will explain you the light dependent reaction more better way than what I have given there in the tube. Okay. This is write the exam year also 2020 3 A level question number 5 E. Describe the linear electron flow that takes place in the chloroplast during light reaction of photosynthesis. I'm going to read the answer. We'll write as point format, put numbers and write it as a list, okay? Number 1, electrons flow through photosystem 1. You can use PS1, that's totally fine, okay? Or else P700 is also fine. Electron flow through PS1 and PS2. Number 2, and other molecular components Point number 3, embedded in the thylakoid membrane of chloroplast. Thylakoid membranes of chloroplast. Number 4, each photosystem has a reaction center complex. Next point, and light harvesting complexes. Next point, the reaction center complex contains a primary electron acceptor. Next point, in PS1, chlorophyll molecule O, listen carefully Putte, that they have mentioned that there is a pair of chlorophyll A molecules at one place, and now here they are talking chlorophyll A molecule, right? So both ways it's correct. So we will be using what is given in the exam script now. Okay, answer script now. Continue the sentence, not a separate one. PS1 chlorophyll molecule O, P700. That is also known as P700. This is the center, reaction center. Okay, P700 absorbs light of 700 nanometer wavelength. absorbs light of 700 nanometer wavelength. Next one. In PS2, chlorophyll molecule O P680. How do we complete this sentence? Absorb light of 680 nanometer wavelength. Absorb light of 680 nanometer wavelength. Point number 9. From this onwards, you will understand they have not mentioned that excitation and returning back. Just they have said excited and these electrons are accepted by primary electron accepted. That's how they mentioned, right? So we will be continuing like that. Number 9, striking of photons of light. Striking of photons of light on the photosynthetic pigments. On the photosynthetic pigments results in the excitation of electrons from PS2 to a higher energy state. From PS2 to higher energy state. Point number 10. These electrons are accepted by the primary electron acceptor in PS2. Next one. Splitting of water occurs due to next point, an enzyme catalyzed reaction. Point number 13, which yields oxygen, which yields O2. Next point, H plus. Next point, and electrons. Next point, number 16. These electrons, within brackets, released as a result of hydrolysis, close brackets, neutralize excited PS2. Emanattam excited P680. Else you can write P680 plus the meaning would give excited. Write all three because we need to know these three names. Sorry? In the exam you will be getting marks any one of these. Okay? Next one. Striking of photons of light on pigments results in excitation of electrons from PS1 to a higher energy state. From PS1 to a higher energy state. Point number 18. These electrons will be accepted by the primary electron acceptor complete the sentence in PS1. Number 19. Excited electrons at PS2 within brackets at primary electron acceptor of PS2 close brackets pass through an electron transport chain. Next point to PS1. Next point, and neutralize the excited PS1 else excited P700 or else P700 plus. Next question, next sentence. Now to the same sentence, write while also. Next point. 22. Energy released due to passage of electrons from higher energy state to, I'll read one more time. Energy released due to the passage of electrons from higher energy state to lower energy state results in the synthesis of ATP. Results in the synthesis of ATP. Tell me one word for that. Photophosphorylation. Put a slash and write photophosphorylation. Are Next one. Excited electrons at PS1 excited electrons at PS1 within brackets at primary electron acceptor of PS1 close brackets pass through an electron transport chain pass through an electron transport chain and pass through an electron transport chain and next point reduce NADP plus reduce NADP plus. In the tube there is in the print I have given as NADP. Please make sure that you have changed that in the tube. Okay, NADP plus. Next one, next point, point 26. To form NADPH. To form NADPH. Next one, which is catalyzed by NADP plus reductase. Got it? NADP plus reductase. Now read the complete thingy. The question that coming up is asking what is happening in the PS1 and PS2. No, we are going to take the points from that into the answer. Okay, so read it well. So I'll be asking which point can be taken to the answer. Okay, we'll take numbers wise. So read it properly. So for the question, you have to pick these points and rewrite your answers. Alright, shall we write again? Write the question and write the answer. Events which occur in P680, P700, those two questions, answers should come from these points. Line electron flow, cyclic electron flow. If you still don't understand, tell me, I can quickly revise one more time. Online students also, if you want, you can request via a message. Are you finished? Nanda. Nakagale. Only that two questions I asked to answer. Okay, don't write all the answers now. Alright, shall we? Now, the questions from light dependent reaction is over. So, we didn't do any MCQ questions. We will be going to the light independent reactions, right? So, are you all okay with light dependent reaction now? Where it is happening? Thylakoid membrane of chloroplast. I'm asking light dependent reaction now. Correct? What are the products of it? Oxygen, ATP, NADPH. Correct. So, what are the two photosystems involved in light dependent reaction? Photosystem 1 and photosystem 2. After absorbing of light energy, what will happen to the photosystems? Results in excitation of electrons. These excited electrons are accepted by primary electron acceptor. Then they will be going through electron transport chain. There are two pathways that we discussed: linear electron flow and cyclic electron flow. What are the things that are not produced in the cyclic electron flow? Oxygen, NADPH are not produced. What is produced? ATP. Okay, now? Hurry. So, we are going to see three pathway of photosynthesis. Right. Now you children tell me what is the location which this reaction is happening? Chrome of the chloroplast. At the There's another name for C3 pathway of photosynthesis. We call it as Calvin cycle. Calvin C. Capital, it's a scientist's name. That scientist is the one who have found out most of the reactions in this particular pathway, so that they have named it after that particular scientist, so that you can't put it in simple words, right? Alright. So here the carbon dioxide fixation is going to happen with the aid of products that you produce during the light dependent reaction. What products will be used in this one? ATP is used. NADPH is used. Alright? Okay. So here carbon dioxide. How many carbon are there? One carbon, isn't it? So this is reacting with RUBP. I write the long term also. RUBP is equals to ribulose bisphosphate. Alright? RUBP has 5 carbons. Okay. So 5 carbon. This will give immediately a 6 carbon unstable compound. Okay. Okay. This 6 carbon one is not staying for longer time period. It will soon broken down into 2 molecules of 3 carbon compound. So here 3PGA. This is having 3 carbon. 2 molecules of these will be formed. Alright? Then this is having 3 carbon. No? This is the first stable product of this particular pathway. Since this is having 3 carbon, we call it as C3 pathway. Understood? This one then converted into 1,3-bisphosphoglycerate. Using the ATP that produced in the previous light dependent reaction. Okay, 1 minute. ATP converted into ADP plus phosphate here. Alright. So then 1,3-bisphosphoglycerate is converted into G3P. G3P is glyceraldehyde 3-phosphate. Do you remember glyceraldehyde? Where did we learn? Trioses, no? Isn't it? Yes, carbohydrate trioses. In that lesson, I told you that you will meet these compounds in these lessons, no? Isn't it? So here I forgot to write PGA. 3-phosphoglyceric acid. Phosphoglycerate also they are using 3-phosphoglycerate. Oh. Right. So here Actually, there is a usage of NADPH produced there. NADPH is converted into NADP plus H pluses plus electrons. Got it? Here, removal of H pluses meaning that this compound is oxidized. Simultaneously, this compound is reduced. Can you understand? Alright? Okay, understand that one. Then, some of the G3P, this is the first first carbohydrate precursor. This is the one which is used to make all the carbohydrates, glucose and all the other carbohydrates. So, some amount of these going to produce carbohydrates. Understood? Then, some of these will be going to regenerate RUBP. There also we will be using some ATP produced in the light dependent reaction. Got it? Hi. So in this pathway there are three main steps. The Calvin cycle has three main steps. So from here to here, what do we call? Number one, carbon dioxide fixation or carbon fixation. Alright. Then here 1,3-bisphosphoglycerate conversion to 3-GP. What do we call? Reduction. You saw that how the reduction is happening. This compound is oxidized. Simultaneously, other compound is reduced. And here the third one, G3P conversion into RUBP. What do we call it as? Regeneration phase. So number 3 is regeneration. Right. Now you guys tell me how many steps are there in this pathway? 3 steps. In these 3 steps, what are the 3 steps? Number 1, carbon fixation. Number 2, reduction. Number 3, regeneration. Hari. So what is the first stable product of this pathway? 3-PGA. So that's why we call the pathway as C3 pathway. Tell me another name for this pathway. Calvin cycle. Why it is called as Calvin cycle? Scientist Calvin found out many reactions of this pathway. In the carbon dioxide fixation, what's happening? Carbon dioxide is taken by RUBP and it's converted into 6-carbon unstable compound. This reaction is catalyzed by a special enzyme called Rubisco. You have to remember this name. Rubisco means RUBP carboxylase oxygenase. Got it? RUBP carboxylase oxygenase. So I'm going to tell you another important thing. This pathway actually has to occur 3 times to produce 1 G3P molecule for carbohydrate production. So I'm going to multiply these numbers. Just read it once and familiar with the given numbers. I'll be erasing all the unnecessary lines meanwhile. So I'm taking 3 carbon over here, carbon dioxide over here. In order to capture 3, how many RUBP should be there? 3 should be there. So here 3 into 5, 15 are there. 15 plus 3, 18 are there. So 18 should be divided by 6. So how many unstable compound are formed? 3. Right. Then how many 3 PGA? 6 put there, right? Okay, because 3 carbon, no? This one 6 carbon, this is 3 carbon. Got it? Right. Here 1 3 bis possible rate 6. That is also having how many carbon? 3 carbon. There's no loss of carbon here, right? And G3P having how many carbon? Again a 3 carbon compound. Okay. So how many of these should be produced here? 6 of these should be produced. Remember, 5 of these are going to regenerate RUBP. Meanwhile, 1 of these is going to produce carbohydrate. Actually, this is having 3 carbon, no children. So that can we produce a glucose molecule from this one? No. So what do we do? This should be multiplied by 2 to produce 1 glucose, isn't it? So that cycle should be happening how many times in order to produce 1 glucose? 6 times. Can you understand? Here only 1 is taken. 2 of these should be there to produce 1 glucose. Understood? Hi. Let's write this one in the given space. Can you go? There I have given it, but I want you to draw it in a paper nicely in bigger version. Don't write any unnecessary things. You can write the key word key at somewhere, and then this one also write it bigger. Write it there, but all things should be there in the tube. That is easy for you, right? When you are studying, if everything is at one place, it will be easy for you. Otherwise, at the exam, you will be in trouble finding where you wrote that one. If the red color ones are not clear, please let me know. Done with it? Right. We'll quickly go to the description given there. Listen carefully. There are little changes to be done. Carboxylation or is carbon fixation. The carbon dioxide acceptor is 5-carbon sugar ribulose bisphosphate. Alright, that is the carbon dioxide acceptor in this particular pathway. RUBP takes a carbon dioxide molecule and forms 6-carbon unstable compound. 1 carbon dioxide at a time. This breaks down immediately into 2 molecules of 3-PGA. 3-PGA is the first stable product of Calvin cycle. This reaction is catalyzed by enzyme RUBP carboxylase and oxygenase enzyme. And actually the 3 points from the next one has to be taken to the carboxylation phase. Each molecule of 3-PGA converted into 1,3-bisphosphoglycerate molecules using ATP produced during light-dependent reaction of photosynthesis. Then 1,3-bisphosphoglycerate are reduced into, actually 2 points from the next one, put an arrow there to the carboxylation. First 2 points. There's the tute. Go to the tute. Page number 5 from the beginning. Beginning, go to the note. 2 points from the reduction phase, take it into carboxylation phase. Then what happens? 1,3-bisphosphoglycerate is reduced into G3P, no? Isn't it? It says then 1,3-bisphosphoglycerate are reduced into G3P. G3P will act as precursor for carbohydrate synthesis. Every 3 molecules of carbon dioxide enters to the Calvin cycle. 6 molecules of G3P are produced. Therefore, the net synthesis of 1 molecule of G3P, make it as for the net synthesis of 1 molecule of G3P, cycle must take place 3 times or thrice. All the above steps are catalyzed by specific enzymes. Even if we don't learn, there are enzymes catalyzing each of these reactions. Okay. Next one, regeneration of RUBP or carbon dioxide acceptor. RUBP is regenerated by undergoing a series of complex reactions. This process uses energy from ATP generated in the light-dependent reaction. Understood that one? Hi. Now everyone go to the wait. Yeah, there are questions from C3 pathway in the structured assay area. Question number 14, 13. Structured assay. Can you all quickly answer so that thereafter we can go to the paper discussion? Those who don't have the tute, please collect the tute from the class. Please take screenshots while I am scrolling down. Finish? Hold on. Right. First question is to draw that one. So you can draw the same thing. Mm, wait. What is the cellular organelle and the location which C3 pathway of photosynthesis takes place? Location is stroma of chloroplast. Organelle is chloroplast, right? Right. Next question. Name the instances where ATP produced during light-dependent reaction is utilized in the light-independent reaction. One is carboxylation, or else you can say that 3-PGA conversion into 1,3-bisphosphoglycerate and regeneration of carbon dioxide acceptor. Next one, what is the amount of carbon atoms present in the first stable product? 3 carbon. What is the name of carbon dioxide acceptor in C3 photosynthesis? RUBP. What is the first carbohydrate precursor in C3 photosynthesis? G3P. What is the use of NADPH which is produced in the light dependent reaction? Reduction of 1,3-bisphosphoglycerate into G3P, or else reduction is also enough. What is the fate of G3P? Some amount is going to generate carbohydrates. Meanwhile, most of them are going to regeneration of RUBP. Right? What are the 3 major steps? Carbon fixation, reduction, regeneration of RUBP. Next one is a question that you can't answer. Actually, the atmospheric carbon dioxide enter the leaf via the stomata. After the stomata, that is going to substomatal cavity. After that, it is going to the leaf mesophyll cells. Just write it and keep. You will understand more this one after 4th lesson. Okay. Amasha and Dilki, I actually couldn't mark your online paper. You can take your paper while I'm reading through the answers. You can mark your answers, MCQ answers. Pute. Amal is absent, no? Amal last week was absent, ne? Savino, that boy, ne? Yeah. Yeah. Rashida. Hi. Okay, shall we discuss children? So I'm going to the paper, 1 minute. So let's discuss. Give me 1 minute. I'll come back. All right. Come to the first question. The nucleus of an organism contains 12 chromosomes. After this cell divides by mitosis. The emphasis is there, 12 chromosomes, it's mitosis. How many chromosomes would be present in one of the daughter cells? It should be 12, no? Mitosis is not changing the number of chromosomes, right? So if you don't understand the concept, that's the concept behind. Mitosis is not going to change the number of chromosomes, right? Next one, which stage of the cell cycle shown in the following diagram? Answer is, it's metaphase 1. Can you all see that it's located on the metaphase plate? Homologous chromosomes are there. Since homologous chromosomes are there, you can identify it is as the metaphase 1. Correct? Why it is animals there? Aston spindle is there only in animal cells. So you can write that. What happens as a result of meiosis? Haploid cells are produced. That's the answer. Correct? Number 4. Which of the following events occurs in meiosis 2? Separation of chromatids. Okay? If we go through one by one, crossing over in prophase 1, replication of DNA in the S phase in the interphase, preparing of homologous chromosomes, prophase 1, having the chromosomes number in prophase 1. Understood? Did we write meiosis essay? Meiosis essay? No. Cell cycle essay we wrote, ne? Did everyone submit? What about you, Shaili? Next question. Select the correct statement regarding smooth endoplasmic reticulum. Missing? Alright, yeah, I have missed. Yeah, okay. 3 more questions remaining, no? Sorry. 2 daughter cells produced in the end of meiosis 1 when compared to the parent diploid cell will have. Answer is half amount of DNA and half the number of chromosomes. For example, at the beginning, if we had like this, I'm taking one pair. We have 2 chromosomes and 4 DNA, isn't it? Neither? Am I correct? It's parent. Parent is it interface one they are talking about. We go to interface. We have 46 chromosomes, 23 pairs. Oh, answer is this. Here, then after that you will receive a cell which is having only 1 chromosome. Now here 1 chromosome, 2 DNA, isn't it? Half of amount of DNA, answer 3. Half amount of DNA and half amount of number of chromosomes. In the process of mitosis, a eukaryotic chromosome consists of go to the table that we did put there for meiosis. Meiosis table also we did now in the class. In the interphase cell, the number of DNA is 2, neither. 2 is converted into 2. We can say that it's not changing also, ne? I have to think about that. Okay, so for now, I'm taking third one as the answer, but I'll think again whether we can consider answer number 2 also. Okay. Next, number 6. In the prophase of mitosis, eukaryotic chromosome consists of 2 chromatids. Um, what is the structure of a single chromatid? Asking about one single chromatid, like this. Having one double helix of DNA coiled around protein molecules, answer number 3. Okay. 7. Which of the following statements regarding both TEM and SEM is correct? Generates 3D images only in SEM. That's why it is wrong. Okay. Next one, select the correct statement regarding smooth ER membrane-bound enzymes present on it. Number 9, select the incorrect statement related to the nucleus of eukaryotic cells. What is the incorrect one? Third one, nuclear matrix regulates entry and exit of substances. What is regulating? Entry and exit, nuclear envelope, nuclear pores, right? Next. Fifth question. The eighth one or ninth one? Fifth answer. No, no way. Red blood cells, there's no nucleus. So, wrong, no? Hari. The common feature of feature it should be, ne? Common feature to both mitochondria and chloroplast is ribosomes are found in both. Next one, 11 and 12. Actually, I took it as 11th question. 12th question, all is given for everyone. 11th question, answer is functions of lysosomes and endoplasmic reticulum is answer number 5, D and B. D is removal of residual materials from cells by exocytosis. B is production of steroids. Sorry, I couldn't scroll the paper. So we are now in the question number 9. 12. 12 all answers given. Next one, in some diploid organisms, haploid cells are produced by meiosis. The diagram shows an animal cells at various stages during the first division of meiosis. Answer number 3. Okay, 3, 2, 4, 1, 5. 14th, which of the following is incorrect regarding cell wall? Some components in the cell wall are synthesized by smooth ER. Yeah. Nitrogen containing polysaccharide. So usually we are chitin. No, sorry. Cellulose is not a derivative. I got it wrong. Third answer. Yeah, in that way it is wrong. Ne? But the other one is mostly wrong. That is Golgi, no? Yeah, cut the derivative part, polysaccharide only. That third one, polysaccharide. RNA, okay. 15th, which statement is incorrect regarding plasmodesmata? Answer number 5, plasmodesmata are common to both living and non-living structures. No, plasmodesmata are living connections between the plant cells. 16th, which statement made by student regarding above ABC structures cannot be agreed with? Answer number 1, A, B, C are subcellular structures found in all eukaryotic cells. Why is that wrong? Some cells are not having mitochondria. For example, red blood cells are not having mitochondria, right? Okay, next one, which is not a difference between prokaryotic and eukaryotic cells. Answer number 2, the cytosol of all prokaryotic cells doesn't have a semi-solid gelatinous mass. Every eukaryote is having cytosol, no? Next one, which of the following incorrect regarding the nucleus? Answer number 5, nucleolus located within it contains DNA is wrong. RNA and proteins can be there. Next, 19th, which of the following phases of the cell division chromatids separate and move towards the opposite poles? Anaphase of meiosis 2 and anaphase of mitosis. Answer 2. In meiosis, synaptonemal complex is formed in prophase 1. During usage of scanning electron microscope, answer number 5, specimens are applied with heavy metals prior to observation. Question number 22. Which of the following is common characteristic to all the cells? Presence of semi-fluid jelly-like substances called cytosol. Answer 3. Question paper is not anyway hard, okay? You could have scored more if you have concentrated on questions. Another. Next one. Select the incorrect statement about cell junctions. Again, similar kind of question was there before. Plasmodesmata, non-living, that's so wrong. It's living connection. So answer 1. Which of the following features differs mitochondrion from a chloroplast? Absence of membrane system inside the matrix. 2. 25. 25. Two incidents that occur during mitosis of the cells are given below. Answer number 3, prometaphase and anaphase is the answer. One part of the question is missing, no, anyhow. So you have identified the question, no? Movement of chromosomes back and forth, that is prometaphase, elongation of the cell in the anaphase. Meiosis is important in creating variations. Which stage is directly important in this regard? Prophase 1, metaphase 1. Prophase 1, the crossing over. Metaphase 1, independent assortment is happening. Hari? Tanashri, what happened this time? In study? It's not only meiosis, mitosis, no, other questions are also wrong. Then you have to study more. There are so many capable students, but at the end, some are not working properly because they are not continuing the hard work throughout. Remember very well, you can. Everyone in here in this class, they can do well, but still, you have to have the courage to continue what you started. Another. Next one. Which of the following is correct regarding history of the cell? Answer 3. Virchow showed all the cells arise from existing cells by cell division. The stage given there is metaphase. Q is metaphase. Okay. The given subcellular structure is centriole. Next one. 31. 30th question. Which cell structures contain nucleic acid? Answer 4. The electron micrograph shows part of a plant cell which is following combination is correct regarding the given labels. Answer 4. Cell wall, vacuole, chloroplast, nucleolus. Next one. Image in the question 31 is taken from transmission electron microscope. How do you recognize? Having a 2D image and a magnified image is given there, so that it's not light microscope. You know, light microscope, you can't see these details now. Next one. In which structures are found in animal cells and in plant cells? Answer B, C, D only. Because centrioles are only in animal cells. Prokaryotes different from eukaryotes. Which of the following character makes prokaryote different from an eukaryote? Answer number 5. Which of the following incorrect about cell junctions? Muscle tissue contains tight junctions. No. What junctions are there? Desmosome anchor junctions are there. Right, 35 questions are done now. I'll give you a little time to go through those questions until I start the rest of it. Please go through. Thirty-nine is the highest. This class 37, no? Highest 39 is there. 37, 37, no? Alright. 36th question. Which of the following events do not occur in the interface of an animal cell? A, B, D. Answer 2. 37. Which of the following statement is correct regarding subcellular components? Answer number 5. 38. Which of the following is not a difference between meiosis and mitosis? Answer 3. 39. Select the incorrect statement regarding light microscope. Maximum magnification is not 100 times, 1000 times. Steps involved in preparation of specimen of an onion epidermal peel for the microscopic observation are as follows. Correct order is A, C, B, D. Next, A, B, C, D questions. Still, I can see some children do not know the concept, and there's a printing mistake is also in the paper. Instead of A, B, C, D, 1, 2, 3, 4 was there, no? So some have mistaken that one, but still you could have asked from me. A, B, C, D. If A, B, D is correct, answer 1. A, C, D correct, answer 2. A, B is answer 3. C, D is answer 4. Any other choices? Answer 5. Hari? 41. Which of the following is so are incorrect regarding enzyme site of the location in eukaryotic cell cycle? Answer number 1. 42, answer number 3. That means A, B. 43, let's go one by one. Answer is 5. Let's see what statements are correct. Segregation of homologous chromosomes at anaphase 1. Correct. Fusion of haploid male gametes with haploid male gametes? No. Haploid female gamete actually. So that is wrong. Pairing up homologous chromosomes and formation of recombinant chromosomes it should be occur during meiosis. Yeah. Replication of DNA in the interphase, correct. Next one, random arrangement of homologous chromosomes at the equator during metaphase 1. Correct. So answer is 5 because 4 answers are correct here. Glycolipids are synthesized by which of the following organelles? Answer should be 5. Lysosome is not synthesizing. Mitochondria is not synthesizing. ER and Golgi. It should be A, B, C, D, not E, F, G, H. Okay. Yeah. G, H it should be. Huh? 45. Which of the following functions is so are the functions of extracellular matrix? Answer number 1, A, B, D. Fourth one, answer number 4, C, D. 47, answer number 5, cyanide binds to at least one molecule involved in producing ATP. If a cell is exposed to cyanide, the most of the cyanide will be found in the places where the ATP is synthesized. No? So, mitochondria is synthesizing ATP, isn't it? Any other answers? Yeah, only mitochondria, no? Next one. Both membrane-bound enzymes and ribosomes are found in answer 4, C, D, mitochondria, chloroplast. 49, select the correct statement or statement. Answer 2, A, C, D. Which structure function pair is mismatch? Nucleolus 1 correct. Lysosomes, extracellular, intracellular digestion also done. Ribosome, protein synthesis done. Golgi, protein trafficking, wrong. Microtubule, muscle contraction, correct. Only one answer is correct, so answer 5. Okay? Yeah. Yeah. Diploid or haploid. If you are using parent cell as diploid, result also diploid. If you are using haploid cell, result is also haploid. Mitosis, both haploid and diploid can be used. 38. Fifth one, the haploid cells are produced. It's a difference, no? Fifth one, haploid cells are produced in meiosis, correct, no? Haploid or diploid cells are produced, that is correct, no? Correct, no? Please go through one by one. I also may have made some mistakes, okay? So if you have anything, please make sure you will ask those things, right? So these question papers are improved once it is being done. One set is doing and they are telling the mistakes and then you are also telling the mistakes and these question papers are improved more and more for the next batches, right? Okay. So how was the paper? Is it good? Okay. Please go through one more time, okay? Hurry. So Amar Shah and Delki, can I know your numbers, MCQ numbers? Put the MCQ numbers here. I'll be looking into that. Yeah. Question. 47 there. Can't mark two answers. In any way, if you mark, yeah, I know that you have to write the correct number of the answer, right? I know that you are correct, right? Not wrong. Because following instructions is important in the exam, okay? I have asked to underline. Okay, okay, that's fine, that's fine. Next time you don't do that mistake. And this one also have done something wrong. Hi, please get used to the question papers. Do more papers. Go through past papers, and then only you will understand this one. Okay. Hari, tell me that one okay no? So we can go to the lesson again, no? Hari. Hari. So we are going to talk about photorespiration. Photo means light. So there's some relationship with light, this process, right? And the respiration, you're making carbon dioxide and water no, another. So this is actually related to Rubisco enzyme. Rubisco, what's the long term? RUBP carboxylase oxygenase. That means this can catalyze two different reactions. That means it can catalyze carboxylation and oxygenation. So in the carboxylation reaction, you know what really happens there. RUBP takes carbon dioxide catalyzed by Rubisco will give 6 carbon unstable compound. It will soon broken down into 2 molecules of 3-PGA. Do you remember? Hi. In the oxygenation reaction, RUBP reacts with oxygen catalyzed by the same enzyme, but it will give 1 molecule of 3-PGA plus 2 carbon compound. There's a name for that, but they have removed the name so that you don't have to memorize the name. So I'm not going to tell the name also. I may have given it in the tube, but still we are going to change it into 2 carbon compound. Hi. So you can see that this is the one which is going to produce 1,3-bisphosphoglycerate. Thereafter, this is the one which is going to produce G3P. G3P is the one which is producing carbohydrates, isn't it? So if the number of these are reduced, number of carbohydrates produced also going to get reduced. It is halved here. That means because of this reaction, 50% of the yield is reduced. Okay. So this process is a wasteful process. Why is that? Production is lower. Remember very well, these two are acting as a competitive substrate. What's the meaning of competitive substrate? Both can join to RUBP and do the reaction. Both are fighting. When carbon dioxide is higher, carboxylation reaction is happening. When the oxygen is higher, oxygenase reaction is happening. This is usually happening in C3 plants in hot, dry climates. Why is that? In the hot, dry climates, plants are closing their stomata to do what? Hmm? Yeah, to reduce the transpiration. Then what will happen? Carbon dioxide is not taken to the leaf. Oxygen is produced as a result of light dependent reaction. So in the substomatal cavity, more oxygen than carbon dioxide. So that which reaction is promoted? Oxygenase reaction is promoted so that these are produced. Remember very well, this compound, if it is accumulated, it's toxic to the plant. Another. Therefore, we are using an alternative pathway to convert this two carbon compound into carbon dioxide and water, which is not toxic to the plant. That particular pathway we can say photorespiration. Here we are using enzymes in three different organelles. What are the three organelles? Shall I write it here? Chloroplast, mitochondria. Yes, got it? This is a wasteful process and also this is an energy demanding process. That means in order to convert this one, you need ATP also. Understood? Let's go to the description given there. Come to the tute, everyone. Rubisco is capable of catalyzing two distinct reactions. 1 minute. Two distinct reactions acting as both carboxylase and oxygenase. Carbon dioxide and oxygen are competitive substrates. Therefore, carbon dioxide inhibits oxygenase and oxygen inhibits carboxylase reaction. High carbon dioxide inhibits oxygenase reaction. High oxygen inhibits carboxylation. Here I have given the two equations. Can you all cut 2-phosphoglycolate in the oxygenase reaction and write their two carbon compound? You don't need to know the name. Okay. In carbon dioxide fixation, two molecules of 3-PGA are formed. Oxygenase reaction results one molecule of 3-PGA and again change one molecule of two carbon compound. It says again, two carbon compound do not have immediate use in the Calvin cycle. Also, in higher concentrations, it is toxic to the plant. It says that processing of 2-phosphoglycolate into non-toxic product in a metabolic pathway using the enzymes in chloroplast, mitochondria, and peroxisomes are called as photorespiration. It is a wasteful process. It consumes ATP. This happens in C3 plants, leads to net loss of carbon, makes 50% less 3-PGA than it would have done if CO2 had been used, reduces yield by 50%, lose productivity, eliminates the net gain of photosynthesis. Photorespiration is a protective mechanism for plants. That means if the compound is there, it is toxic. It is going to remove that compound in a metabolic pathway. Remember very well, since this process is kind of reducing the yield of photosynthesis in C3 plants, In the evolution, we assume that C4 plants have evolved. That you will understand once we're done with that particular reaction. That plant, this reaction is not going to happen. There are so many mechanisms to prevent this one. Okay. It says why photorespiration occurs in C3 plants on a hot dry day. Most plants close to most plants close. There's a repetition, no? Close plants again. Most plants close their stomata in order to conserve water, not test, right? Water to reduce transpiration. At the same time, oxygen released from light reactions begin to increase, and this lead to further reduction of carbon dioxide to oxygen ratio in the cytosol. This leads to photorespiration. In order to solve this problem, new photosynthetic pathway, as I told you, C4 is evolved in order to overcome this problem. Got it? I'll take a little time and study about that one. And I want to give that meiosis question also. I can remember I gave like, okay, I'll give it again. Write that and come as a homework when you're coming on next day. Please make sure that everyone is submitting. Nowadays, some are skipping. This is 2022 A level, question number 5. Okay. Describe the nuclear division where the chromosome number is reduced during meiosis. Read the question and understand the question first. First of all, read and tell me what they're asking from us. What are they asking? To describe which stage of meiosis? 1 or 2? Oh, quickly tell. Hmm. Okay, Dilki. Okay, Amarsha, right. What do you think? Meiosis 1 or meiosis 2? Asking to describe. If we can remember the table, you can give the answer quickly. What is it? Which one? Rashida? Go to the table. Quickly go through the numbers that I have asked you to highlight. We did the table for meiosis as well, isn't it? I didn't discuss, ne? First one, meiosis 1 is asked to describe. Can't you understand? Can you? I'll quickly go to table also if I have asked you to do it and come. Write the question and keep it as a homework. Did you all write? I'm erasing. Uh, Cell with 6 chromosomes. Number of chromosomes. Did I write chromosomes first? DNA. 46 chromosomes. Human cell. Chromosomes. DNA. Interface only I gave, no? G1 is G2. Not like that, no? Interface only, no? So you have to write what is happening at the end of interface, okay? So if the 6 chromosomes are there, DNA number is double, 12 it should be in it, isn't it? After space, it should be 12 now, isn't it? Am I correct? Correct or wrong? This one interface, usually it has like this, no? That is also not visible as a chromosome, as a network of chromatin, but still, for 6 chromosomes are there with one single DNA here. But in the space, it will be double, no? Arda? So 46, 92. ඊළඟට prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis. Prophase 1, nothing is happening. Anaphase 1 also, sorry, metaphase also, nothing is happening. And anaphase 1, things, homologous chromosomes are separated, it's same, no? Isn't it? Telophase 1, again, same number. Cytokinesis 3, 3, 6, 6, 23, 23, 46, 46. So if you highlight this one, you will understand chromosome number is half. So the question is asking you to describe this one, first part of meiosis. Then we'll go to prophase 2 and metaphase 2, anaphase 2, telophase 2, cytokinesis. Right? So prophase 2, you will get, I'm taking one cell having 3 chromosomes, 6, 23, 46 here. Metaphase also same number, 46. Next one, metaphase, sorry, anaphase. Now it's going to change. Here is the place that you are going to change. Number of chromosomes would be double because chromatids are separated. Is it okay? Right? So chromosomes would be separated. Chromosome number is double here. Neither. Anaphase I'm talking. 6. DNA number is not changing. Here is 46. Here is 46. And the telophase 6, 6, 46, 46. Again dividing into 2. Here it is 3, 3, 3, 3, 23, 23, 23, 23. That question is a bit problematic because question number 5 I'm talking. Uh, at the end of meiosis 1, that means cytokinesis 1, it's asking half the amount of chromosomes, no doubt. The DNA number would be halved only if you consider after S phase. Before the S phase, the chromosome number would be same nේද? Parent cell එක අපි S phase එක undergo කරලා නැහැ කියලා ගත්තොත් අර ගන්න පුළුවන්. So I'll rethink about this and I'll get back to you, okay? Are you done? Shall we quickly go to the next part also, C for photosynthesis? Erasing. That day, what did you write and give it to the class? Cell cycle. I didn't collect it from the class. Hi. So C for photosynthesis. And that is happening in two different cell types. This one cell I'm drawing. This first cell we call it as leaf mesophyll cell. This is called bundle sheath cell. If I draw it and show you, if you take a plant leaf, there is an epidermis. No, this is mostly monocot plant, so there is no distinguish like palisade and spongy mesophyll here. So this is the cuticle. So here having bundle sheath cells, which is covering or enclosing the vascular bundle. Can you understand? And here is xylem. Here is phloem, right? And this area is filled with mesophyll cells. So in this one, actually, they are having tight connection between these mesophyll cells. They are arranged like a flower wreath. Right? Okay. This is like a flower wreath. The lower epidermis also here. There may be stomata over here. This particular arrangement, we call it as Crohn's anatomy, like a flower wreath. Crohn's. They are tightly connected with the mesophyll cells because they are going to share certain materials which are produced in the mesophyll cells to the bundle sheath cells. They are going to give so that they are connected. Okay. Hi. So I'll label bundle sheath cells. This is mesophyll. Understood? Hi. So here is leaf mesophyll cell, bundle sheath cell. Photosynthesis is happening in both the places. Light independent reaction. So here, actually this carbon dioxide going to get dissolved in water and form bicarbonate ions. I write that equation also here. Give me 1 minute. I get the correct equation and write. This bicarbonate is the one which is taken to this pathway. Bicarbonate ions are taken by PEP. PEP is having how many carbon, children? This is the 3 carbon compound. This is a 1 carbon compound. So what would be the result? Oxaloacetate, which is how many carbon? 4 carbon compound. So now you know 4 carbon, which is the stable product of this pathway. That's why we call it as C4 pathway. Understood now? Right. So this reaction is catalyzed by PEP carboxylase. That one has only carboxylase name. So that there's no relationship with oxygen. This one is very efficient, is capturing carbon dioxide. There's no relative, there's no friendship with oxygen. Okay. Hi. This oxaloacetate then converted into malate. There are plasmodesmata which are connected. There are numerous plasmodesmata connecting these two cells. Malate is diffused to the bundle sheath cell. Malate is then converted to pyruvate. Malate is having how many carbon? 4 carbon. Here is 4 carbon. Pyruvate is having how many carbon? 3 carbon. Pyruvate 3 carbon. Here is 4 carbon. What has happened now? Carbon dioxide is released. Understood? So this pyruvate is then again diffused into the mesophyll cells. Then it will be regenerating PEP, carbon dioxide acceptor. Understood? So remember, this carbon dioxide is now going to get captured by someone we know. Who is that? RUBP. RUBP taking carbon dioxide. Now you know what's going to happen. What's happening? 6 carbon unstable compound. Then it's soon broken down into 2 molecules of 3-PGA. Then converted into 1,3-bisphosphoglycerate. Then it's being converted into J3P. J3P, some amount is going to produce carbohydrates and the other is going to regenerate RUBP. Where did we use ATP? Here, ne? ATP converted into ADP plus phosphate. Alright? And here also regeneration part, ATP converted into ADP plus phosphate. And here this place reduction, no? So I'll be taking it here. So NADPH is converted into NADP plus H pluses and electrons. Understood? And here also we will now. Can you hear me now, Amasha? Right. Should I explain this again? Okay. So here this reaction is happening in two different types of cells. Firstly, it's happening in the leaf mesophyll cell. Carbon dioxide is not taken as the gas itself. It's converted into bicarbonate ions. In the aqueous format, bicarbonate is reacting with PEP to give 4-carbon compound oxaloacetate. It's being converted into malate. Malate then diffused into the bundle sheath cells via the plasmodesmata. Then it's converted into pyruvate while releasing 1 carbon dioxide. RUBP is accepting carbon dioxide and Calvin cycle is happening there inside. Meanwhile, pyruvate is going back to the mesophyll cells and then will be converted to PEP. In these lessons, make sure you always mention the number of carbon when you are writing a compound, okay? Can you hear me now? Hi, I just read what is there in the handout regarding C4 pathway description. Just read it one more time before we end. I added only one thing there in the description. In the sentence, pyruvate is produced. There, within brackets, decarboxylation. That's the only thing I add. Pyruvate is produced, that part, decarboxylation. Right. So, children, we are going to finish the class. So, hope you understood the lesson. We will be discussing more and more about C4 pathway. I didn't finish. Just started only. Yeah, you can have that. Okay. Second part of the paper will be given sooner, but I can't promise that I will give on next week because I'm too tired this with this. I have a lot of work regarding 2024 children. So, please wait until I give the answers. Okay. Right. So, everyone, study well and continue all the good work. Good evening and have a nice day. Bye.